Now we'll continue on part 3.2, where we will be using synteny to gather additional evidence for the ortholog assignment. In the NCBI RefSeq genes track shown in the genome browser, click on the XM ending in 5862. And note that this is the accession number for the Diacuba RefSeq containing the mRNA transcript that is aligned to this region for the Diacuba scaffold. On this page, notice that the position can also tell us where the transcript will start and end. Now, below the GenBank record, scroll down to the bottom of this window to the translation sequence within the CDS section. We are now viewing the computational predicted protein sequence of the putative ortholog to RAP-PA in Diacuba. This sequence is translated from the predicted mRNA. Now we'll copy the accession number for the translated protein sequence. Now you may have noticed that this accession number has a two-letter prefixes followed by an underscore. These RefSeq accession numbers has an implied meaning with respect to the type of molecule it represents. For example, we have a known or predicted model followed by its type of sequence, mRNA or protein. The NCBI database of reference sequences, or short for RefSeq, is a curated, non-redundant collection of naturally occurring DNA, RNA, and protein sequences. The RefSeq database includes both known, that is manually reviewed by NCBI staff or collaborators, and computational predicted sequences. A complete list of the RefSeq accession number prefixes is available in the NCBI handbook. The next step is to navigate to the NCBI BLAST. Click on the Protein BLAST button so we can do a BLAST-P search. Paste the accession number for the translated protein sequence that we copied from the GenBank record for the transcript XM ending in 5862 into the Enter Query Sequence text box. Under the Choose Search Set for the database, we'll select the reference proteins to search. In the organism text box, enter Drosophila melanogaster with the taxonomy ID of 7227. Now in the query sequence, we are looking to match the predicted protein sequence of the putative ortholog to RAP-PA in Diacuba. And the database is our subject, that is a collection of sequences we are searching for matches using the melanogaster reference protein database. Now we'll finish off by selecting the checkbox next to the Show Results in a new window, and then click on the BLAST button. Our BLAST-P search found 45 proteins within the melanogaster reference protein database that show similarities with the protein sequence of the putative ortholog of RAP-PA and Diacuba. However, only one of these is a good hit. The best BLAST-P match to the putative ortholog of RAP-PA in Diacuba is described as the Ras homolog enriched in brain, isoform A, in Drosophila melanogaster with its corresponding accession number of NP730950 with an e-value of 6 × 10 to the negative 131 and a sequence identity of 97.25%. The remaining hits had a much higher e-value ranging from 5 × 10 to the negative 41 to 0.002 and a much lower percent identities ranging from 43.75% to 23.48%. Therefore, the BLAST-P search result shows that the Diacuba RefSeq prediction XP ending in 1796 is most similar to the first hit among all the annotated proteins in the melanogaster. The description of the best hit only tells us the name of the gene, but in order to know the gene symbol, we can confirm that by assessing the accession number of this hit, clicking on the NP730950.2. And now we'll scroll to the bottom towards the CDS feature And the line with the gene equals tells us the symbol of this ortholog, which is Rab. Hence, based on parsimony, the BLASTP search result supports the hypothesis that the XP ending in 1796 is the putative ortholog of Rab in D. jacoba. And now that we have identified the transcript for our target gene, we need to repeat the BLASTP search for the transcripts of the two neighboring genes on both sides of the XM ending in 5862. To help us stay oriented to the location of our target gene while looking at the neighboring genes, we can right-click on the RefSeq transcript of XM ending in 5862, and then select Highlight XM ending in 5862. We should now see this region highlighted in blue, and when we zoom out to examine the neighboring genes, we can easily visualize where our target gene is because it will remain highlighted. You can also highlight additional regions by repeating this process. And to clear a highlight, right-click on the highlighted area and then select Remove the Highlight. When right-clicking on the highlighted area, there's also a Zoom to Highlight option. Now, if the coding isoforms of your gene differ, for example in this region, it is recommended to use the longest one that is best supported by the other lines of evidence as the query for BLAST search. However, choosing a different isoform than the longest will likely still give the results you need. And lastly, in some region of the analysis, you may encounter a nested gene, and that is a gene located entirely within the intron or exon of another gene, typically on the same strand of the DNA. Now, the host gene and the nested gene may be unrelated in terms of function, and they can be transcribed independently. Let's take a look at this example here. Say if this was our target gene, and then the gene overlapping our target gene is the nested gene. So some of the key points about nested gene is location. The nested gene is completely contained within the non-coding intronic region of the larger host gene. And in terms of transcription, both genes have their own promoters and regulatory sequences, so they can be expressed separately. And the functions of the host and the nested gene are often unrelated, but their physical proximity may have some regulatory or evolutionary significance. And that being said, nested genes are found in various organisms, and it may arise due to evolutionary processes like duplication or rearrangement. Now that we have examined the genomic neighborhood of the putative ortholog, we need to identify the direction of transcription for the putative ortholog of RAB-PA and also the neighboring genes in D. jacoba. And then we'll use this information to draw a sketch of the genomic neighborhood. To do so, we need to repeat the process we followed in part 1 and use the strategy described in steps between 9 and 10 to determine the orientation of the putative ortholog and the transcript of the two neighboring genes on both sides. After we draw our sketch of the RAB gene in D. jacoba, we will compare the relative order of orientation of the genomic neighborhoods of RAB between D. melanogaster and D. jacoba. If the genomic neighborhood looks similar between RAB in D. melanogaster and the putative ortholog in D. jacoba, then we can be confident that we have found the best candidate for the ortholog. However, if any of the information is inconsistent with this being a local syntenic region, with local synteny being conservation of the genomic neighborhood, then we should inspect our other hits in the T+N search from part 2 to see if a different genomic region is a better match overall. Now, examination of the genomic regions surrounding the RAB gene in D. melanogaster and the putative RAB ortholog in D. jacoba shows that the relative gene order and orientations are the same in the two species, and hence the synteny analysis supports the assignment of D. jacoba feature at the NC052530 scaffold and located at the coordinates of 19150,510 to 19152,080 as a ortholog of Rab. Keep in mind that if your target species assembly happens to have been numbered from the opposite end of the relevant scaffold, then the orientation of the orthologs could be the opposite of what you see in the melanogaster, but still be syntenic.